Yeah, I'm so sorry that I can't be there. This would be such an amazing meeting to attend in person, but circumstances outside of my control prevented it. So I'm going to tell you about our work on trying to use Stentor ceruleus as a model for understanding how cells learn. And in case I forget later, the work I'm going to show you is the work of some of it I've done myself and also a student, Deepa Rajan, and a former postdoc, Tatiana Makushak. So Stentor is a remarkable cell. It's a ciliate like paramecium, but it's very large, about a millimeter long, dark blue. You can fish them out of ponds all over the world. We actually can get them out of the Golden Gate Park just down the road. The cells have very complex and beautiful structures, which we also study. And it's been a classic model system, for 100 years, not only for studying how cells build complicated structures, but also for how cells do behaviors. And so Jennings, for example, had a lot of studies on Stentor, more than a century ago. In order to be able to harness this powerful and very convenient classical model system, we had to do some of the work ourselves to make it molecularly tractable. So we've sequenced and assembled the genome. It's now publicly available. And we've gotten RNAi by feeding an injection working so we can perturb any gene of interest that we want. And so with these tools in hand, and we're currently developing additional tools, it's time to really dig in and figure out what is the mechanism by which a cell can learn. So habituation in Stentor, there's many examples, I think, now of cells that can learn and behave in various ways. We really like this particular paradigm because it's so simple. So what happens is when the cells are happy and feeding, they'll stretch out like a cone. They filter feed bacteria and algal cells out of the pond water, and they're very happy in that condition, but they're also big and vulnerable. And so when predators like fish or small invertebrate embryos strike at them, they can contract out into a ball in about a millisecond. So it's a very rapid contraction response. But it's energetically costly, so they only want to contract if they're being hit by an actual predator, as opposed to casual contact with other pond organisms like swimming algae. So Stentor is able to habituate so that the cells, if they're bumped with a certain consistent weak stimulus, they'll learn to ignore it, and that allows them to continue to feed when there's not actually a threat. So this is just on the left, some examples. On the left, top left, you see images of Stentor cells when they're happy in their extended form. The cells are about a millimeter long, as I said, so they're quite easy to see even with low power objectives. Then on the bottom, after you tap the dish, the cells all contract down into a ball. Shown in the upper right is a plot of the probability of response versus trial. So this is tapping once per minute, and this is data from a paper by David Wood. We did a lot of work on Stentor habituation in the 80s. So there's a whole series of really beautiful papers characterizing this and really showing how many of the classical features of habituation are met with Stentor. And I think for this audience, I don't need to point out that if people want to complain whether habituation is really learning, I think it certainly is, and because I've been asked that. And I think you have no further to look than the classic aplesia habituation response and all that we've learned from that. So the advantages of studying Stentor habituation as opposed to any other cell behavior that you could study is that it's extremely simple behavior, very easy to score. The cells are either contracted or not. There's no room for personal interpretation. It's a nice binary behavior, so it's easy to write software that can just automatically measure the response. Also, the experiments are very rapid, so you can do the entire thing in an hour, which means that in one day you can actually do many experiments. And because you have cells growing in a dish, you can do a whole lot of cells all at once under identical conditions. So it has a lot of experimental convenient features. The price you pay is that it's this weird model system for which there's very little known at a molecular level, but we're trying to overcome that limitation. So it's been reported that some of the old papers from Jennings that you can poke the cells with a hair and they'll contract. You can squirt noxious things at them and they'll contract to that. And Mike Levin will talk more about, I think, those experiments. And Jeremy Gunawardena will hear more about these sort of stimulating cells with noxious stimuli. The advantage of mechanical stimulus is that it's, again, very easy to control. The problem is doing it with a hair, it's not controllable. And so we wanted to build an apparatus. So in 2012, as part of a course I was running, we built this electromagnet-based setup where the cells sit in a dish on a metal strip, which is a ruler. You have an electromagnet under computer control, and that provides a precise tap. The problem with this apparatus is that it's very hard to, if you want to vary the strength of the tap, you have to move the mechanical stop, which sets how far the metal bar can swing. And that's just not convenient to do during the course of an experiment. So recently we've been working on an improved version where the mechanical force is applied by a stepper motor. Otherwise, the geometry is very much the same, so it's compatible with our previous work. And the beauty of using a stepper motor is you can control the strength of the stimulus in the course of one experiment. So you can give cells series of strong and weak taps and see what happens. So there's a lot more flexibility. So this is just a close-up view of how this works. And so it's very easy to build and to use, and we published on how to do that. So this is the kind of data you get. These are the fraction of cells contracting in a dish versus the stimulus number. It exactly matches what Wood published decades ago. So that's good. I think we're all in agreement about the result. And it's quite interesting to see, actually, we think about single cells learning in this graded way, because it makes you think that maybe a single cell can store many different levels of habituation, which would tell you something about how many states a cell could be. And maybe it's actually a continuous range of states, as we heard from Carl's talk at the beginning. It turns out, though, that this is what we see at the population level, just counting how many cells contract versus stimulus. But when you look at individual cells, what you see is that typically cells will start out contracting to a large proportion of the stimuli, and then there's often a very visibly obvious switch beyond which the contraction probability drops. And depending on the cell, this can happen at different points, and the individual probabilities can be different, so it's all statistical. But we do think there's a visually apparent step-like response at the individual cell basis. And if we measure lots of cells, we have a statistical test where we feed in the single cell data, and then we ask if we check at each point during the course of the experiment, is there a point at which you see a maximum difference in contraction probabilities before and after that point? And then you can do that again. So once you find where you think there's one step, now you can subdivide up the remaining time and ask, is there a second step? And the data you get is plotted here, where we see essentially very strong statistical support for a single step in the response, but not for more than one step. So it does seem like there's a single step taking place. So, and in addition, consistent with the idea of a single step controlling the habituation, if we ask what is the dwell time that prior to that step occurring, they're actually geometrically distributed, which is exactly what you'd expect with a simple two-state model where you switch to a less responsive state with a constant probability every time a stimulus comes in. So this leads to the simplest model we have that we think can recapitulate the data at hand, which is you have two states, a more responsive and a less responsive state, and they can transition between them with certain constant probability whenever in between successive stimuli. Now, with this model in hand, we can try to fit the data that we see and then ask what parameter values you need to do that. And so we can get very nice fits to the population level data using parameters that we extract by fitting a combination of population and single cell data. And we can do that using either moment estimators or using hidden Markov model strategies. We get basically the same result either way. And one thing we've done is to go back to some of David Wood's results, showing that, as with other habituation cases, typically it takes a lot more stimuli to habituate to a high force, whereas to low forces you can habituate relatively more quickly. And so we also see that happening. And then when we fit our parameters to the data, single cell data from those two force regimes, what we find is that the probability of contracting in the responsive or non-responsive state are actually very similar between the two force regimes. The big difference is the probability of switching from the responsive to the non-responsive state. That goes up a lot when the force is low. You also see that the probability of going back towards the responsive state is extremely low. So these are probabilities of a transition occurring in between successive stimuli given once per minute. So this is a very, very long time frame that we're looking at for these things to go back, which means essentially during the course of our experiments, we're looking at an irreversible process, essentially irreversible. So what ends up happening is cells start out responsive, they gradually habituate, and now they're sort of sitting at this non-responsive state. So then the question is, okay, this works for the data that I already showed you. Is this really what's going on? Could it really be as simple as just these two states behaving in a nice, clean Markov process-like way? And as anyone who's done biology can probably guess, no, it's not going to be that simple. But I still think you should start with the simplest model and then try to see what are the ways in which it fails. So one way in which that simple model doesn't work is when we look at really high forces. So we can go to an even higher force training regime. And what we see is that you still see the response grade off. So as you have increasing stimuli, the cells will gradually ignore more and more of those stimuli. But what's different is, and then if you look at individual cells, they also basically still undergo a single step. So that part still holds. But now if you look at the number of stimuli elapsed before the cell takes the step from the responsive to non-responsive state, now it's no longer exponentially distributed. Instead, the step times are error-long distributed, which is what would happen if several events have to happen in a row before the cell can take a step, meaning that there's got to be more than one state transition occurring prior to a visible change in transition probabilities. So this is not something that you would be able to explain with a straightforward two-state Markov model as we described. So whether that means there's some kind of memory going on or just multiple invisible states that we can't see, that's a little harder to determine. And we're trying to figure out some clever strategies to give cells different kinds of input sequences, some strong and weak forces, and see if we can probe it more that way. As one step towards that, one kind of crazy experiment that we did, which was actually forced on us by a reviewer for our paper, and there's a good chance that reviewer is out there in the audience, and if so, I'd like to thank you very much for this suggestion, because I didn't want to do it, and we more or less did it because we were forced to, which is as follows. So Stentor contracts within a millisecond, but it takes about almost a minute for the cells to fully re-extend. So typically, when we vary the interval between the stimuli, we might go to a minute, minute and a half, 2 minutes, but we never really went to much faster stimuli. So what we did in this experiment, though, was to stimulate cells once per second. So that's much faster than they can re-extend. So they contract, and then before they can extend again, you can make them contract again. You do that again and again and again. And if the video is kind of hung, but yeah. All right, I'm just going to jump to the bottom line. The bottom line is that the cells actually habituate extremely quickly when you do this. And so here I'm plotting stimulus number again versus fraction of cells contracting, but now this is stimuli happening 1 per second. And what we're seeing is that before it would take, on the order of half an hour to fully habituate. Now they're habituating within a couple of minutes. And so after a couple of minutes, even though we're hitting them once every second with the stimulus, most of the cells are completely ignoring it. And we can show that they're not exhausted and so on. We have controls for that. So it seems that Stentor actually learn more quickly when the stimuli are coming at a higher frequency. And we can plot this data 2 different ways. So on the top, I'm comparing our usual 1 minute stimulation with our faster stimulation. And what you see is that as a function of stimulus number, the faster stimuli, the more frequent stimuli are less effective. That is, it takes a larger number of stimuli to get you contracted. But as a function of absolute time, the more frequent stimuli work faster. And so the only way to really reconcile all this is to say that somehow the transition probability from the responsive to non-responsive state has to depend on the time interval between the stimuli. So again, this is another deviation from the classic Markov behavior. The probability is not constant in unit time. Somehow something is happening, there's a timer or something where the cell is able to sense how much time has elapsed between the stimuli. And another phenomenon that also speaks to that same thing is if we look about the recovery or forgetting a phase of the cell. So if you give Stentor cells a series of stimuli, they'll wait till they habituate, then you stop stimulating them for some period of time, and it could be half an hour or whatever, and then resume stimulation. It's been known from work of David Wood that on the time scale of tens of minutes to an hour, the cells will fully recover. And we've shown that on our hands anyway, under our setup. If we compare the contraction probability of completely untrained cells, so that's the initial response, the probability of contracting once they're fully habituated after that first set of stimuli, which is called the habituated response on the right. And then if we turn off stimulus for 15 minutes, almost all the cells go back to the fully contracting state. So that means that basically the transition rate from the fully contracted state to the responsive state is happening at a rate of a fraction of a minute. It's very, very fast. And this is orders of magnitude faster than what you'd predict given the reverse probability of transitioning, the reverse transition probability from our two-step model to fit to the actual habituation data, which means that this reverse transition rate is somehow a function, again, of the frequency with which stimuli are coming in. So if they're coming in on a 1 per minute basis, you have a very, very low probability of transitioning backwards. Whereas if their stimuli are coming in less frequently, now you get a much higher transition probability. So one model that we have is that something is changing in the system. One idea we have is something like a watchdog timer. Maybe every time a cell feels a stimulus, even if it's not contracting, because remember, these are habituated cells, nevertheless, it knows when it's being stimulated and it resets some kind of a timer. And only when that timer never gets reset and it realizes there's no stimuli coming in, now it changes to a different rate of the reverse transition. So how that's happening at a molecular scale is, I think, a fascinating question. We don't have an answer for at this point, but I think it's really interesting to think about. So this idea that something different happens when the cell starts to forget, meaning there's a clear change in the rates, got us wondering, maybe something has to happen in terms of building new molecules. And so we started testing what happens if you impair protein synthesis. So if we treat cells with cyclohexamide, one thing we know is it does not actually affect their ability to habituate. So if you poison translation, cells will still habituate just as well as if you didn't. So you don't need to make protein to learn. However, if we compare what happens when we do our forgetting experiment with or without cyclohexamide, what we see is that when the cell, sorry about the noise, that's our apparatus going in the background. What we find is that indeed the cells remain habituated even though they're not getting any stimuli when you block protein synthesis. And this is just quantification of that. Some of the cells do recover, but the vast majority of cells never seem to recover as long as protein synthesis is inhibited. So we also know, though, that as I mentioned, the cells can still habituate when you when you poison translation. They still contract just fine, so there's no effect on the actual motile machinery or the sensing machinery. And once you've habituated them in the presence of cyclohexamide, if you hit them with a very large force, they still respond just like a normal cell would. And so again, it's not in any way impairing their ability to sense the stimulus or to respond. So this is making us think that forgetting is somehow maybe even more of an active process than learning is. This is kind of an interesting parallel, I think, to computer science, where sort of the physics of computation has shown that in theory anyway, you can store data with zero energy cost, but then you have to burn energy when you want to erase data and reuse a memory unit. So maybe there's some kind of analogy there. And there's also, I guess, a philosophical question, which is I've always imagined that learning would be the active thing that requires energy and synthesis and so on. And then forgetting would just be a passive loss of whatever it is that you've learned. It's not obvious to me, though, in the case of habituation, what constitutes learning. In other words, we typically say that cells learn to ignore a stimulus, and then later on they forget that they should ignore it. But I think it's equally possible to say that when the stimulus goes away, they learn that they should be responding to it when it happens, and that during habituation experiments, they're actually forgetting. So if anyone has a really great, crisp, bright line distinction that they can draw between learning and forgetting, I hope they would share that with me or maybe the audience in general, because I think this is a topic that this whole group might have also tackled with. I'm just not equipped mentally to understand the difference. So I just wanted to close by just mentioning a couple of other things that we're working on in the lab, just very, very briefly, just interesting points of comparison. So I think a really key question for understanding behavior of cells or any system, behavior of cells is this idea of a state space. And there's a lot of interest, as I know you all know now, in trying to define cellular state in molecular terms. So you try to measure every transcript and maybe every metabolic state and every mRNA level, every protein modification, and so on. You're going to have, hundreds of thousands or millions of microscopic variables. And the idea is, well, we'll do principal components or something like that, or UMAPs, and try to get clusters. And you can try to do that, and I think that's an interesting and productive way to do it. But the idea there is to have some kind of reduced dimensionality state space. And I think in general, people are finding continuous state spaces where you have this sort of smear of possible states. The other alternative is to use behaviors to try to reconstruct state spaces, where you try to ignore the underlying molecular details and just ask what are the different states a cell can be in terms of the ability of those states to predict future behavior. And then the question is, what do those state spaces look like? Would they also be continuous? Might they be more discrete? And this whole continuous discrete question has a lot of interesting implications. For example, in terms of what happens if you have an infinite number of time steps, are you guaranteed to revisit prior states and so on? But also just in what kind of tools you would use to think about it. So I think if you have discrete states, you can bring a lot of tools from theoretical computer science, think about things like finite state automata, push-down automata, so on. Whereas if you have continuous states, now it's much more dynamical systems, and you want to think about things like Hamiltonians and so on. So we've been trying to look at several different cell types and trying to see which kind of state spaces do you get. So I told you about our work on the stentor habituation, where it really does look like it's something like a two-state model, although it's probably more complex than that. So now we're trying to figure out is there a more complicated underlying space going on there. We've also been looking at walking gait in a Protus euplotes uristomus. This is done by a postdoc, Ben Larson. And our idea there was that we know that, so these are ciliates that walk using clusters of cilia called cirri. And it's known for animals that coordination of gait involves neuronal circuitry. And here we wanted to know, could a single cell without a brain still coordinate the gait of its walking? And the answer is yes. You see actually there's more than two states, so it's a more complicated state space, but there are very clear, discrete, distinct states, and only a small fraction of the possible states. In other words, there's 14 different feet that could be walking at any point in time. Only a small fraction of combinations are ever used. And the transitions among them are quite non-random. Some of them violate detailed balance, and so there's cyclical behaviors and a lot of interesting dynamics going on there. And we have evidence originally from chemical perturbation and now from RNAi experiments that it's the microtubule cytoskeleton is involved in regulating these state transitions. So we think actually euploidies may constitute some kind of mechanical computer, which would be really quite interesting. Then when we try to look at, I guess, more conventional model systems, namely mammalian cell types, a former student, Amy Chang, was looking at the dynamics of how internal organization of a cell could change looking at mouse embryonic fibroblasts. And so she measured a number of different descriptors of organelle structure. And the result there was that you see a continuous state space. But if you look at transitions within that state space between, first of all transitions occur between adjacent points in space. So it's a continuous motion through this continuous state space. And if you look at the probability of transitioning from one state to its neighbor or back, it turns out to actually obey detailed balance, which means that you can represent in principle, you can represent the dynamics using a single scalar potential. And remarkably, when she did that, indeed, that potential function, so you just take the occupancy of the states, use that to produce a potential. And it turns out that then the gradient of that potential actually does predict the future behavior of the system. So I think in that case, maybe it is really tractable using sort of these very simple classical physics tools. And then another student, Jacob Kimmel, was looking at the motility of a number of cell types, including mouse muscle stem cells, and their behavior is really in classifying their trajectories and using that to define what state they're in. There again, he sees a continuous state space, but now detailed balance is violated, and there's really quite rich and interesting dynamics. And so the question now is, what actually is the law of motion within the state space of kind of a generic cell? One concern that I have with all this mammalian stuff that we've done is we're always looking at cells grown in a dish, which is not their native environment by any means. And I've heard it said that studying behavior of a human cell in a dish is like studying behavior of a human inside a burning building. And I think there's probably something to that. So I do personally think we have a lot, we're going to see more, we're going to get better representation of actual behavior when we look at naturally free living single cells like the protists we've been looking at. So I just want to close and acknowledge the people doing the work, especially I mentioned Deepa Rajan's work on stentor habituation, started originally by me and then Tatiana Makushak, and also Ben Larson working on the euploides walking behavior. And we've had a lot of really great collaborators who are still helping us work on these things. And we've been extremely lucky. I mean, that agencies have actually funded this work. So NSF in particular, NIGMS, and also want to thank the Fondation Formentère Gilbert for giving us their I2 Cell Award to support our study of finite state machines and cells. And I will stop there and take any questions.
Okay, thank you very much, Wallace. I'm going to ask the first question, and that is you make this distinction between passive learning and active forgetting, and so but the memory you're looking at, the type of learning, doesn't last for that long a time. So what I'm curious is if you have looked at memory in stentor that lasts for, say, 24 hours, and then you see if that depends upon protein synthesis, and based on all the other work, one would predict that it would. So in that case, you'd see a distinction between the active and passive learning.
Yeah, so we are starting to do that. So we've started to do 24-hour, 48-hour experiments. We've also been doing an experiment where you habituate them for an hour, turn off a stimulus, let them recover, then do it again, and then do it again and again and again, and ask, superimposed on that repetitive behavior, is there a longer time scale change? And this is something that Deepa was really starting this spring. I think will be a focus of the work this summer. So yeah, we're very much on board with that. I will say though, there's plenty of examples where protein synthesis is affecting cells in a huge way on the same time scale that we're looking at. So I don't think it's insane. But personally, I do think probably protein phosphorylation is more likely molecular substrate for memory if you were going to pick one. I was kind of surprised actually by the result that we got with cyclohexamide at all.
Yeah. Thank you. Okay, there's a question in the back. Would it be possible to get a microphone? Great. Thank you, Eric. Thank you.
Thanks. I wanted to point out that there's a whole line of research in cognitive psychology that sees forgetting as a key part of learning. That it clears out the irrelevancies that you're exposed to every day, keeps you from remembering the unnecessary, and that relearning after forgetting will be more efficient than the initial learning was. So I think what you're seeing is very much in line with what's going on in that research with forgetting and learning as just being two sides of the same coin.
Fantastic. Yeah, I will have to delve into that. But it also makes me think that what we should do is now ask, do those cells that were habituated, fail to forget, will they be able to, if they eventually do recover their behavior, will they now not be able to rehabituate because the failure to forget has screwed up their ability to make a new memory? Are there any particular authors that you would suggest or keywords I should look for?
Well, the mic went away.
Oh.
Sorry. But I'll relay it.
Okay, forgetting as a part of learning.
Robert and Elizabeth Bjork. Here
At UCLA, they're in the psychology department.
Perfect. Thank you so much.
Yeah. Okay, Hassam,
Hi. Can you hear
is
me?
it on?
I don't think so.
I can hear
No, it's
you.
on. Okay, cool. So I really like your approach of like trying to explain behavior in terms of the most simplest model with the least number of states. Do we know if the habituation to mechanical agitation transfers to other types of agitation, let's say noxious chemical stimuli?
We know from David Wood that it does not transfer to habituation to light or electrical shock. I don't think anyone, we have not done it with mechanical stimulus. Jeremy, are you in the audience? Jeremy Gunawardena there? He may have done this.
He's here. He says he hasn't done this.
Okay.
Yeah.
Okay. But that would suggest more states than two because they need, at least a pair for every type of stimuli, right?
Yeah, no, that's exactly. And this also kind of gets to, well, it suggests that there's more states than two, and that each of those states might be some, multi-bit number that tells you whether or not to respond to stimulus X but not Y. Yeah, it'd be super interesting. I fully agree.
Eric?
If there's no other questions and there's time.
Is there a remote question? Maybe we'll take the remote question. Eric?
So Ollie asked, you showed faster habituation with higher frequency stimulus. Did you ever test different intensities of stimulus along with increased frequency? That is, do you get faster habituation with the less intense stimulus if you increase its frequency and even faster habituation if you increase both its intensity and its frequency, or is the frequency the most important parameter?
That is such a good question. I will say we have not yet exhausted all the possible combinations of frequencies and stimuli. For the high frequency, I think we've so far have only done a force regime, a sort of moderate force regime. We have not tried extremely high forces at high frequency. I think we might have tried that in the cell. So one frustrating thing here is that when you hit cells with too much force, they actually swim away and you can't finish the experiment. So we try to avoid getting to that. And I think that was happening more easily with the high frequency. But I actually think looking back, we gave up too soon because I think there was actually people with too high of a force in, and it was actually smashing the whole apparatus to pieces. So I think we do need to go back and retry that. The closest thing to what you're saying, Ali, is we tried swapping out every other high force stimulus with a pair of low force stimuli applied at a higher frequency. And the result there was pretty wild, which was that if you watch the responses, and you actually, which response was to which of the two kinds of stimuli, what you saw was that the pairs of high frequency weak stimuli showed a rapid fall off in their response, whereas the individual high force stimuli showed a much slower fall off. So it was as though the cell was somehow tracking two different responses, which again gets to the suggestion that I think the previous question was that maybe there are a lot more states than we really are seeing. And we're just setting up our experiment in such a way that we only happen to be interrogating one state at a time, which is a great thing to do from sort of a quantum physics point of view, but the reality may be way more complicated than that. So what we want to do now is go back to some of these things people do in neuroscience, where we can, with a separate motor method, we can basically inject white noise and give random sequences of all kinds of frequencies, gaps, and intensities, and then get to a lot of those, and then try to back out by reanalyzing the data post hoc what the actual stimuli were that were driving which kind of response.
Very, very interesting. Okay, we were going to have to move on. So thank you very much, Wallace, for a really interesting
Thanks to the audience for these fantastic questions.